Hi, my name is Elaine, and I'd like to thank you all for coming to my presentation. Before we get started here, I'd like you all to take a quick survey. The URL will be here. It's basically this question: if you could pick the traits of your future child, would you? So getting started. On August 6, 1945, the first atomic bomb was dropped on Hiroshima, Japan, causing thousands of deaths and devastating destruction. I just told you my topic was on genetics, so why exactly are we discussing this? It's simple, really. Once the knowledge on how to create such a weapon was released, it could never be taken back. We are still affected today. 9 nations around the world have nuclear weapons, and there are 16,300 in total. And if one were to launch an attack and another a counterattack, we'd have mutually assured destruction, meaning basically both would die and we could have the end of the world as we know it. Recently, scientists have found a tool that could change humanity's future, but for the worse or for the better. One thing's for sure, once this technology is used to edit our genes, it will change the world forever. Let's just hope the result of nuclear weapons doesn't foreshadow the possibilities of genetic engineering in human beings. To ensure that this never happens, I have come to the conclusion after a year of research that only certain areas of genetic engineering in human beings should be explored and tested until they can be safely applied using modern-day science, while other areas should be banned entirely to ensure humanity doesn't become too wrapped up in its own evolution. So first today I'm going to give you like a brief science lesson on genetic engineering and the different categories. Next we're going to talk about current events. Where is genetic engineering now? What is this new technology that's been discovered? Why is there so much hype around it is what we're going to discuss after that. What can it do for us? What does this mean? Next we're going to talk about why certain things might stand in the way, morality being the main objection. And lastly, we'll talk about alternatives to genetic engineering. So, before we delve into a moral debate, let me explain exactly what genetic engineering is and the different categories you need to know. Genetic engineering can be defined as the editing of an organism's genes in order to manipulate a certain trait. We see the results of genetic engineering every time we step into our local grocery store in the produce section. So you see a stand of tomatoes, and they're huge and vibrantly red. You can almost be assured that they did not grow naturally that way. Scientists manipulated certain traits specifically to make the tomatoes appealing to consumers. The two categories within genetic engineering are somatic cell gene therapy and germline cell gene therapy, as you see these two bubbles here. This just means there are two different types of cells DNA can be taken from to edit. So what's a somatic cell? A somatic cell is any cell in the body that is not a reproductive cell, which are eggs and sperm. Editing the genes of a somatic cell affects only the individual the changes are made on. This is really important and will come into play later, so I want you all to remember that. Next is germline gene therapy, and the germline cells are eggs and sperm, they're reproductive cells. And editing the genes of a reproductive cell will not only affect the individual the changes are made on, but also their entire progeny. So their children's children and the children's children's children's, so all their descendants. When learning about recent issues in genetics, a phrase that you will commonly encounter is genetic enhancement. It should be noted that genetic engineering and genetic enhancement are different. Genetic enhancement is taking the methods of genetic engineering and using them for things other than medical cures. It is the idea of improving traits such as height, strength, intelligence, etc. Oftentimes, genetic engineering will be referred to as a slippery slope, because people believe it will start off with good intentions, trying to cure hereditary diseases, but will eventually become a tool we use excessively for the unnecessary. For example, like changing the eye color of your child so that they'll have blue eyes instead of brown. To me, human genes editing human genes sounds like some fictional dystopian future that is thousands of years ahead of us. So why are we talking about it now? We are specifically talking about it now because a new technology was discovered called CRISPR-Cas9, which enables scientists to cut and paste sections of DNA more effectively and more accurately than ever before. This means that the future I thought was thousands of miles away could actually be just around the corner. Here's a quick video to explain what CRISPR does from its very own founder, Jennifer Doudna.
A few years ago, with my colleague Emmanuelle Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists...
All right, we're going to stop there. And in case you didn't catch it, one thing I really wanted to highlight is that on April 18th of last year, a study was published from a group of Chinese scientists stating findings after testing human embryos with CRISPR-Cas9. So she briefly mentioned that some scientists have already started testing this on human embryos. Although the embryos were non-viable, meaning they couldn't eventually become babies, a debate was still sparked by the public. Some thought an ethical boundary had already been crossed, but we will get into that much later. What they really found is that CRISPR isn't ready for the advanced measures they would like to take, which leads me into my next point. I didn't mean to alarm you, and if I did, I'm sorry. The truth is that CRISPR really isn't ready for use in humans right at this moment. When tested in complex organisms, it tends to have unpredictable results. In this specific experiment, the scientists attempted to use CRISPR to remove a gene that causes a fatal blood disorder. It was performed on 86 embryos. Out of that, only 28 had the gene cut and removed correctly, and out of that, even fewer replaced it with a new healthy gene. Eric Lander, a geneticist and professor of biology at Massachusetts Institute of Technology, wisely states in his article called Brave New Genome that it has only been about a decade since we first read the human genome. We should really exercise great caution before we begin to rewrite it. The scientific field focused on DNA and genetic engineering is incredibly fast-moving. Look how far we have come in genetics research just within a decade. But this also alludes to the fact that we still have more to learn about DNA itself. It is hard to correctly edit and manipulate something you don't already fully understand. A hashtag was even made to toe down some of the hype that CRISPR was getting after a Wired article came out on the topic that really concerned a lot of people morally about the subject. It's called hashtag CRISPR Facts, and basically, just jokes were tweeted about CRISPR to kind of tone down the hype. Like, CRISPR really isn't ready for use, so we shouldn't really be concerned about it. Is basically what they want to that's the message they were trying to send. But one thing that we should note is that CRISPR may not be ready for correcting human deficiencies yet, but it could be in the near future, so we should talk about ethical boundaries now so that we're prepared. And with that, we'll move on to the hype. Why is it even worth pursuing? Why are we talking about it? What are the positives in this? And really, it's because CRISPR-Cas9 could end up being a cure-all, basically. The main reason scientists are even examining gene therapy is the hope that it can cure genetic diseases and help us gain a better understanding of more common diseases. Genetic diseases like sickle cell anemia occur because of a defect in one gene. If CRISPR-Cas9 could be used to alter this gene, we could have a cure for disorders like this one, and Huntington's disease and cystic fibrosis. Editing germline cells of people afflicted could remove the disease from humanity's entire gene pool, so we never have to deal with it again. Another exciting hope for the use of CRISPR in humans is that it will help us make connections between certain genes and diseases like Alzheimer's and cancer. Before CRISPR, this process was a slow form of trial and error and of testing in animals. Now the process can be sped up and connections found much faster. However, these harsh diseases may contain roots in hundreds of different genes, making it more difficult to foresee whether gene editing could actually offer a cure. And lastly, another thing that genetic engineering could promise, more like genetic enhancement, but it's still a viable positive, is that it could create a human race that could think quicker, run faster, climb higher, and feel deeper than ever before, and could lead humanity to stretch beyond its physical and cultural limitations that are biologically in place now, to become more improved in almost every sense of the word. By using genetic enhancement, we are reaching humanity's full potential. So we could use it for the betterment of our human race, basically. And so now, what's holding us back from using this right now? And basically, it's morality. There are lots of objections from bioethicists and also the public about how using this technology and editing our genes is morally wrong. But there are also some moral supports, which we'll get to. One of these moral objections is that people fear that we'll have a God complex. So to take the step of engineering our own genes is to take human evolution into our own hands. It is essentially playing the role of God. Our species would be making monumental decisions on which genes in future generations survive and which die out. Genetics would never be up to chance again. This can go one of two ways. It can make a race of superhumans, or create a future dystopia where genetics is used as a weapon in the form of super soldiers. So, as you can see, this is risky. Also, a concern is a loss of freedom. So if selecting children's genes ever becomes a reality in the future, not only genes for physical traits would be determined, but also genes for personality traits. A large part of the human experience is finding and defining oneself as an individual. If a parent picked out its child's personality traits, this journey of self-discovery no longer holds the same meaning. I'm sorry, that was loss of individual. Loss of freedom is next. The children subject to genetic engineering may feel as though they have lost freedom to create their own identity as an individual. The child's freedom of choice would be detrimented. Genetic enhancement decisions made by parents would end up steering their children towards certain life choices. And lastly, another moral objection would be a loss of equality. So, genetic engineering, if it ever came into being that it would choose certain traits of babies it would be quite an expensive procedure. So the thought would be that a new distinction would be formed between the rich and the poor, because only the rich would be able to afford to genetically engineer their babies, and so a divide would become between natural humans and enhanced humans. The one moral support I have on here is actually from a professor. I can't remember where. I'll add that into a presentation note. If we had the technology, she says, if we had the technology and knew that it was safe, it is our moral obligation to make our children the best that they can be. So I'll let you decide which you side with, but before we go into that, we should talk about alternatives to genetic engineering. So one of the main appeals to genetic engineering is that a lot of parents are concerned about having healthy children, especially if they have recessive genes for dangerous genetic diseases. So if that is the case, don't worry, you can still possibly have a biological child. Adoption is one of the alternatives, but another way to have a biological child is to try PGD screening, which basically screens embryos that have been fertilized outside of the body from you and your spouse, and they find the embryo that is least likely to inherit this genetic disease, this genetic gene that you presumably are passing on. Another alternative is third-party reproduction, so having a sperm donor or an egg donor, and the last is adoption. So to put my knowledge into use this year, after a year of research, I decided for my service learning to write an editorial for the Highlander. I'm submitting it to the Highlander, which is the school's newspaper, and that will be coming out in May. I'm also going to try to submit it to the Eccentric, if you guys know it, which is the local Birmingham newspaper. So if you guys have a subscription to that, please keep an eye out. But basically what it will be on is to advocate for my opinion on to ban germline engineering. So basically I talked about how some parts of genetic engineering should be explored while other parts should be banned to ensure our safety earlier. Now I'm going to get more specific since you guys know all the background. I support somatic cell gene therapy, but I think that germline gene therapy should be banned because of the risks. Remember that editing germline cells means the changes are passed down to every descendant. This means no mistakes can be made. Edits would stick for forever. Although the rewards of gene editing are tempting, the risk that comes about because of germline editing puts a damper on the excitement. This doesn't mean I think the field should not be explored and tested and more. I fully support the testing and editing of somatic cells. This way, some benefits can still be achieved while risking a lot less. So if you guys would like to learn more about that, feel free to read my editorial. And lastly, I'd like you to answer the question you answered at the very beginning. If you could pick the traits of your future child, would you, now knowing what you know now? So I'd also like if anybody changed their mind to raise their hand. Thank you so much for listening to my presentation. If there are any questions, please ask now.